Hello, my name is Carolyn Mendoza. I'm a third-year student majoring in chemistry here at UC Merced, and I'm here today with Janice Guillory, who is a lecturer within the chemistry and biochemistry department. She is the instructor for the general chemistry labs. That's Chem 2 lab, Chem 10 lab, and she's also an alumni of UC Merced, part of the inaugural class of 2009. So, Janice, I will start off with an easy question. Are you looking forward to next week's experiment?
Oh, definitely. Next week's experiment is actually one of the funnest experiments for the Gen Cab.
Very colorful.
Gen Lab, yeah.
Yeah. So I think we have some samples here. This is the inorganic pigments lab. So students use these droppers that we have behind us, and they mix together different compounds and write down qualitative observations of the color changes they observe. So that one is lots of fun. We're speaking now during spring break, so it's like the halfway point of the semester. What experiments have your students been doing so far? What topics have they been learning about?
Okay, well, a couple experiments that we've done, especially the first experiment, because we want to get these students kind of get their feet wet on what they've been learning. So especially with the lab setting, right, where they get familiar with the glasswares and everything, and they learn how to use them correctly.
Yeah.
So like balances, they're able to learn how to use. Yeah, exactly. How to tear it, meaning zero it out before weighing something. They've learned how to use some of the pipettes that we have, and then these cool burettes that we have as well.
I kind of want to zoom out a little bit and talk about kind of the importance of laboratory courses to the learning process. There are a lot of students who take Chem 2 lab, chemistry majors, biology majors, I hear even the applied math majors.
The engineering majors as
well.
well.
Yeah, so a lot of folks take this lab in particular. And I know for this semester there are 30 different sections for Chem 2, which probably is a headache for you.
One of the biggest classes that we had so far.
Yeah, and each section has a capacity of 24 students, so 24 times 30, that's 720 spots, yes. A lot of students. So I guess as from the instructor's perspective, what are like the major takeaways from Chem 2 lab that you want students to have? Like what experiences do you want them to have in this course?
Well, I already mentioned how they should be able to develop their laboratory skills, right? Not just for chemistry, but science in general, because you did mention biology. So one thing that's good about it is that, you know, especially for our chemistry class, we do align the concepts that you learn with any hands-on experiments that we have.
Yeah.
So it actually does give the students a deeper understanding of the ideas that are presented during lecture, right?
Yeah.
Because there's one thing to just kind of like hear about it, and the biggest thing is actually being able to do it.
And I think something that came to mind while you were saying that was Hess's law, or the calorimetry experiment that you do at the end of Chem 2, where you're thinking about thermochemistry. And I remember during lecture that was really abstract to me.
Yeah.
I was like, wait, are we adding this? Are we subtracting these terms? But doing the experiment with the little coffee cups, that really helped me learn.
Yeah.
All right, so since we are here in the room already, I was wondering if we could maybe look around and kind of explore some of the equipment that we have.
Okay, great.
All right, so we have a lot of equipment here. Some of this might look unfamiliar to students who have never been in a lab setting before. Why don't we just talk about, I guess, what the standard set of equipment is for a group of students?
Yeah, right. So as you can see, there are two chairs over here, right? Usually when we do our lab experiments, we want our students to work as partners. This is what a typical lab station would look like, right? So we have these hot plates over here. We have some of the glassware over here that we use. Beakers are always common, right? Now, these are called graduated cylinders, right? So we have a small one, a medium one, and a large one, right? These are actually the ones that we use to measure liquids before we pour them in either a beaker or a flask. Mm-hmm. Flasks are good when you like swirling things. Now, this one's actually pretty cool because it is a hot plate, right, where we can heat things up. It also doubles as a stirrer. When we heat stuff up, and especially if it's something long where you would have to like heat it up for like maybe 30 minutes or an hour, you don't want to constantly stir it by hand. Right, so what's cool about this one is that we do have what's called a magnetic stir bar, where because our hot plate has a magnet as well, a magnetic field. So what happens is that if we drop our stir bar here, and then we turn this on, it actually stirs the solution for you.
Something that was new to me when I first came to UC Merced was having access to fume hoods inside of a lab. In my high school, we didn't do that many labs to begin with, but we for sure didn't have a fume hood. So could you maybe explain why we would want to work in a fume hood and kind of the additional safety it provides?
Okay, great. So fume hoods do have, like, I don't know if I open this one, I don't know if you can. No, you can't really hear, but there is supposed to be air ventilation on this end. I think one of the things that we've done recently was we added aluminum with a strong base called potassium hydroxide, and then adding those two chemicals actually generate some hydrogen gas. And we know hydrogen gas is highly flammable, and we do not want them around the lab itself. So the fume hood actually contains the hydrogen gas in it because of the good ventilation on it. For the knobs over here, you know, we have an air knob, we have a vacuum knob, and one is a gas knob. So it depends on what we want to be using. The gas one, we actually use it when we have our Bunsen burners out and we want to light a flame on it. For the vacuum ones, we have a setup called the vacuum filtration, where we use the vacuum to suck up the water faster from a solid product. And for the air, we mostly use this to dry our glassware if we want them nice and dry before we when we do our cleanups.
So now we're in the organic chemistry, the first semester organic chemistry lab room. After you complete the general chemistry courses, second year chemistry students move on to studying organic chemistry. And as you can probably see already, this room has a lot more going on in terms of new instruments and new equipment that you have the chance to practice with. So Janice is going to walk us through some of the new equipment here and give us an idea about how we can use them during a synthesis reaction.
Alright, so this is called a melting point apparatus. So what this does is if we have any solids that we need to figure out what the melting points are, we'll be using this one. Now, this is a capillary tube, right, where we put the solid in. So you just need a really small amount like that. And then there's a little slot in here that we're going to be putting the capillary tube in, because this melt temp apparatus actually heats it up, right? So then if we turn it on, over here, there should be a magnifying glass somewhere here so you can actually see when the substance does the melting point, right? So then, and then you turn it up, and then you can also track the temperature that it's melting at, right? And then once it's all melted, that's going to be the melting point.
Yeah, and melting points are important for characterizing a solid. If we have a product and we want to see how pure it is, we can measure if it has the correct melting point that corresponds to what's published out there.
Yeah. Great. This one is a glassware kit. We definitely have like different types of glassware in here. The most interesting part would probably be this one. It's called a separatory funnel, right? It's used to extract some organics with some water-soluble things. So you guys remember what like oil and water would look like, right? So they do separate it.
Yeah.
So the separatory funnel actually does kind of like the same thing, where you want to separate something that's either water-soluble to something that's not by pouring out or dispensing out the water-soluble stuff. Okay, so vacuum filtration, just you know, just like any, because we use filter paper, right? And then usually we use gravity to separate solids and liquids. But if we want, especially if that solid actually does trap a lot of water in it, we want it to probably do the filtration a little bit faster than just using gravity. So what we need is this filter flask here, because we actually have to, you know,
connect it to this
the
pump
hose
over here.
to the pump there. And then we would need what's called a Victor funnel. It has the rubber stopper over here because once we turn on the vacuum, it's gonna suck up the funnel into the flasks to create that vacuum system.
Yeah.
And then so then we pour in our solid, liquid, slurry mixture, right? And then we turn on the vacuum, and then it should suck up the liquid out of the solid, like in like 5-10 minutes, and then it does ensure our solid to be nice and dry.
Alright. One cool new instrument that Chem8 students get to work with is called a rotary evaporator, or rotevap for short. Janice, when would we want to use a rotevap in some kind of a synthesis reaction?
Okay. Well, since we are in an organic chemistry lab, usually we deal with a lot of organic chemicals now. So whenever we have, say, an organic product that we need, and then it's obviously dissolved in an organic solvent, or it's mixed in with it, and we want to purify our organic compound that we need to actually evaporate out the solvent, so we can get a nice purified product at the end.
Should we remind our audience who might not know what the technical meaning of organic is?
So organic just means that it's not water soluble. That's it.
Yay!
For Gen Chem labs, or general chemistry labs, we use a lot of water soluble solutions. For organic labs, we use organic solutions, which are the opposite of water soluble solutions.
Well said.
Okay.
Alright.
Alright, so what we want to do here is we have our, say we have our solution here with both organic compounds that and then what we're going to do is first we're going to put it in here.
Oh, we don't have a cat clip, huh?
No, that's okay. Now this is a water bath. So this water bath actually will be warm enough so we can actually, because it does help the solvent to evaporate out faster. And we use this lever over here to lower it into our water bath, and then this acts as a stir, so it does have the temperature constant all throughout our flask. Yeah. And then once everything is nice and set up, we usually turn on the rotevap. We're not going to do it for this purposes, but yeah, so that's when it, this isn't a vacuum system actually, because it does, one, make the temperature, or at least like have the solvent evaporate faster as well. And then what it does is that the evaporated solvent, this is called a condenser, it condenses out to this
collection
flask, yeah. And then it's usually thrown into a waste bottle, and then once everything is done, we should have our nice purified product.
Awesome! Alright, that is all we have for you. Thank you, and look, I hope you enjoy your labs.
Yes, it's gonna be fun, no matter what.